Adhesion junction. Adhesion junctions are a link between cells to make tissues. So the cells that are linked together are mostly adjacent from each other, and once they're linked together, or they work as one unit. There's a several proteins that I'm going to talk about in the next slide. Um, there are two types of attachments and adhesive junctions. There's the adheron junctions and the desmosomes. So since there are two types of attachments, there's several different structures. With the adheron junctions, you have actin microfilaments that you can see on the right in the top figure. Then you have the desmosomes, which are intermediate filaments or the tonofilaments. As I said, there's quite a few proteins that are involved in these junctions. And if you're looking at the figure on the right, you can see the majority of them. So the proteins that are involved are the cadherins, the beta-catenin, alpha-catenin, P-cadherin, E-cadherin, and P120 catenin. These junctions are mostly found in epithelial cells, such as cells lining the intestines, the kidneys, or the epidermal cells. They make a continuous belt that encircles the cell near the apical end of the lateral membrane. They also have multiple functions, such as initiating and stabilizing of cell-to-cell adhesion, regulating actin cyto- of regulating the actin of the cytoskeleton, intracellular signaling, and transcriptional regulation. I was having issues finding diseases that are associated with this malfunction, but what I did find was just mostly generalizations of mutations that happens in the adheron junctions or in one of the proteins. So the first one was a mutation with the adheron junctions that was in the plakoglobin in individuals with Knox's disease, and the second one was human adheron junction gene mutation with a dermatological prevalence, which involved the P-cadherin. One of the papers that I found was talking about a lot of different junctions, like tight junctions, gap junctions, and adheron junctions. So this paper, I just focused on the adheron junctions and what all went into it. So this is just the importance integrity of the cell-to-cell junctions for this cell. So disulfide bridges played a pivotal role in this experiment for the function of the linkages or bondage of the cell-to-cell adhesions of epithelial cells. So what it showed was that cell-to-cell junctions are important for providing efficient barriers against the environment. It was also showing that it is stable, has a stable mechanical connection between adjacent cells that permits the layer to be exposed to external mechanical stress without losing the cell's integrity. One of the things that they added to the cells was Z H1, which basically um, separated the cells from each other, so it kind of broke the bond. But like the alterations to the morphology of the adheron junctions was so small that it really didn't affect the cell as much as it did with the tight junctions, the gap junctions, and I think it said the desmosomes as well. But the adheron junctions really wasn't affected that much. So my question on the topic is, like I said, like I searched some, but I guess I didn't do enough searching. But I was just wondering if any of y'all know what diseases are associated with the malfunction of this junction. Um, if y'all can find out, please let me know.